next speaker will be Chetan Poudel from the Department of Physics and Astronomy at Oberlin College. He worked in the lab of Yumi and Jiri there, and they have collaborators at Cambrian Technologies and the Department of Biomedical Engineering at the Cleveland Clinic. And the title of his talk today is Use of an Inverted Linear Hallbach Array for Magnetic and Size-Based Separation of Magnetic Nanoparticles.
So my talk is Inverted Linear Hallbach Array for Separation of Magnetic Nanoparticles. This is the Hallbach array. Before I get into that, I'll speak about what nanoparticles are. So nanoparticles are extremely tiny particles. They're about 1/1,000,000th of a millimeter in size and are of very significant interest to the scientific community because they act very differently from bulk matter of the same type and have a very high surface area to volume ratio, which makes them very useful for biomedical applications like targeted drug delivery or cancer therapy by hyperthermia. So the way it works is that magnetic nanoparticles have this biocompatible shell, which can be loaded with certain drugs and then injected into the body for these uses. But there is a great need to ensure uniformity before we inject these particles, and that is because we need uniformity to know how much drug delivery we are doing in the body or what temperature we're heating up the cells to. It also allows us to optimize how much volume we're injecting into the body and thus reduce toxicity because these could be toxic. So there's various separation methods that have been devised for gaining uniformity in these particles. Most of them have been targeted towards more larger micron-sized particles. There's also some lab-on-a-chip devices that a bunch of labs have been doing all around the US or in the world to sort individual nanoparticles. But these are really slow because you're sorting individual nanoparticles on the basis of size. There's some more crude and small-scale separations that are more magnetic and size-based sorting of nanoparticles, but our research is based on more of a characterized and well-characterized magnetic separation. So our approach is to take this Hallbach array, which is a series of linear permanent magnets with the magnetization vector rotated for each magnet by 90 degrees. And this gives us this nice feature where it's a one-sided flux. There's a high flux on one side and there's a low flux on the other side. So it's kind of like a refrigerator magnet in that sense. But the main feature here is that we get very uniform field and gradients over the planes that are parallel to the surface of the array where we can do these separations. Most of the research groups have been working with this high flux side for various applications like maglev trains or something like that. But we're using the low flux side, which we haven't found in the literature that nobody's used. It gives a very tiny field, and that is just right for selectively pulling down certain particles, but not others. So if we have a mixture of bigger and smaller particles in solution, we can selectively pull down certain particles while letting the others flow freely. And this combination of a low field yet a high field gradient allows for the separation of these suspensions. So based on our approach of a Hallbach array, we constructed a device and we modeled it to work a certain way. So we have a modeled prediction, which is the red curve, and the blue curve is Gauss meter probe measurements to see that our constructed device agrees with our model. And we also find that the gradient is pretty high according to our model, which is what we need. We need a high gradient for magnetic separation. So we also made a separation channel after we agreed that this array is working as it should. And these are the components which, when sandwiched together, we have a separation channel. There's a tubing that can go through there and there. So, okay, so this is the inlet, and this would be the outlet where the nanoparticles flow in and out. And in between, the separations are occurring. And these happen at a certain distance above the array. So the distance dictates how how much magnetic force is being applied to the particles that are flowing in there. So that can be increased or decreased by increasing the number of components or the number of plates in there. So the mechanism of this separation is this interplay between two different energies. There's a random thermal motion that comes into play when we're talking about these very small particles. They tend to go whichever which way. And there's a dipole-dipole energy term for these nanoparticles can be taken as dipoles, so they interact with each other. And there's a dipole-dipole energy term that scales with m here, the magnetization of the nanoparticles. So what we want is, we want bigger and larger particles with a higher magnetization to cluster together, because if they have a dipole-dipole energy term that is bigger than this random thermal motion, they will start clustering and coming down and settling. Whereas in the smaller particles, we want this thermal fluctuation or the kVt energy to be higher, so that should dominate this so that they don't cluster together. And for 5 and 20 nanometer particle mixture, this is an experimental parameter, we found that we should be at a height of 0.3 centimeters, and the speed and gradient should be this high, where we can see 20 nanometers starting to cluster and the 5s starting to flow free. There's also some forces that we need to take into account. There's a magnetic force being applied by this magnet array down here, which is pulling down these particles, and there's a drag force when they experience that in fluids. So these two counteract each other to give us a drift velocity of motion, and that allows us to find a flow rate which we can use to inject fluid into this system. And this flow rate, we've found out that it's pretty high throughput compared to lab on a chip devices or other sort of techniques. So the mechanism here in another schematic is that these 20 and 5 nanometer mixtures come together, and wherever the gradient is high, since the magnetic force depends on the gradient that I showed you earlier here, these larger particles start to settle down and cluster because of the dipole-dipole energy term being bigger than the thermal motion, and we can see these regions where they start clustering, whereas we don't want these smaller and larger particles interacting. So the small particles are just flowing through, and we get this out here, and all the bigger particles will be trapped in that channel. There are a few complications that could come out in this. So there could be bigger particles that are less magnetized, and so they never start clustering. So we could have some contamination that way. And there could be smaller particles that start interacting with the cluster, so that there's also contamination here in the residue. But for experimental parameters, it seemed to be working pretty well in terms of, we see these banding that we expect from this picture. We see these very localized places where there should be bands and there should be clusters, so that we see that. And we can characterize, after we inject particles, we can take the filtrate out and have a residue. We can withdraw that residue and take different scans with magnetometry, with transmission electron microscopy. And our special technique is to use a small angle X-ray scattering using an X-ray diffractometer, which is a pretty widely used technique for characterizing this particle size. And we use a Rigaku Ultima instrument for that. So to characterize how efficient our separation has been, we use this small angle X-ray scattering. This is the instrument that we use, and this is the data that we've got. We know that small angle X-ray scattering, the intensity patterns vary with the particle size and the particle distribution, so that by looking at this pattern, we can know what sort of particle size and what distribution it is by fitting the data. So the data that we got here, this is the 5 nanometer stock solution that we ran through. This is the 20 nanometer mixture. Then we mixed these two together and got this blue line, which is a 50-50 mixture of these two different particles. And once we flown it through the channel, we got this out. This is the filtrate, which is closer to the 5 nanometer. So this is something that we expect or desire. We want the smaller particles to be in that filtrate, not the larger particles. We see that a 50-50 mixture can yield something like 80% 5 nanometer by volume. in a single pass when we're taking it through. So there's still some contamination of about 20 nanometer particles there, but in a single pass, we've eliminated quite a bit of those. So in conclusion, we have successfully constructed this device that can sort nanometer particles magnetically and also based on their size. We've characterized separation efficiency using our small angle X-ray scattering technique. And we recently published a paper based on our findings, which just came out in July. So the future work is in determining what controls the sorting ratio and if multiple passes can improve this result. And we're also planning to work with particles of different sizes and different moment values and to design and test a new channel that we've been working on. So I'd like to thank all the NSF grants that I've been working with, and Bill Martin for machining the early prototype, and Professor Yumi Jiri, who's my research advisor, for guidance.
This talk is open for questions and discussion.
So about how good do you need to get in terms of the purity? I would imagine 20% is still too high for the ones that you don't want.
So in terms of getting commercial sort of nanoparticles, they seem to have a really wide distribution. And we want to get as close of a distribution as we want. 80% distribution is still pretty good in terms of just having a single pass through. But it's just a matter of increasing that percentage to a higher percentage, I think. There have been clinical trials done in certain cases, but the better idea that we have about how tight we have that dispersion, then the better idea we'll have of how much drug delivery we're doing. So that's the thing.
So you talked about how these nanoparticles are magnetic and you take advantage of that to separate them. Is there any other reason for them to be magnetic? Is that necessary for drug delivery or any other biological function, or is that just for the separation?
So in terms of hyperthermia, I was talking about a certain application of cancer therapy. So the way it's used is these nanoparticles are magnetic. They have a certain antibody binding to it, and they can go target a very localized area of the body where there's a certain antigen being overexpressed. And so they bind to that certain tumor cell only. And what we can do is we can control this from the outside where we cycle through a high field, and these start heating up because of the small hysteresis that they have. And so having magnetic particles allows us to heat them up and kill the tumor cells. And that's why we need magnetic particles in that application.
Mike, did you have a quick question?
Yeah, I was curious about what type of chemicals you use for your stock nanoparticles.
So we have just gotten commercial particles. There's ways to synthesize them in the lab,
Yeah.
but we haven't synthesized them. We just use commercial particles. So these are iron oxide nanoparticles that are mixed in toluene, which is not exactly the solution that you use for biomedical applications. There's also, you can use iron oxide in water. That's the most widely used for biomedical applications, but this is stable suspension and it works the same way as water, so we're using those and characterizing. So our future work is in using water samples and other sort of different samples to see if it works in the same way.
Okay, what kind of differences do you expect by using water?
So in water, there's a special problem of it not being that stable in suspension. Sometimes they start crashing out. In toluene, it becomes much more stable. So that allows us to have the nanoparticle stop for a longer period of time to do our test runs on. But there should be issues in some crashing out, some separation, some aggregation in there. So we expect that if we're working with those. There's another group in our college that's working with those particles to work with ovarian cancer. So they've been encountering that sort of problems, some aggregation in water-based particles.
Very good. Let's thank our speaker again.